right, hello Kristin, thanks for your talk. So you were talking about high throughput methods for estimating food contact substance exposure. Can you tell us what was the main message of the talk that you just gave?
Right. So the US EPA is required to prioritize thousands of chemicals for further study. And in order to do that, we have to look at exposure via many different routes or pathways, through consumer products, through industrial releases, and the food packaging pathway is important. And so we developed high throughput methods to include that pathway in our predictions in order to make decisions about chemicals.
You were also talking about the uncertainties that occur in your approach and about ways of quantifying them. What we say, what are the biggest challenges that we are facing when it comes to the quantification of uncertainties?
Well, a lot of times in high throughput models, because we're applying them to thousands of chemicals, we have to make reasonable decisions about input parameters, and that imparts a lot of uncertainty. What we do try to do is then quantify that uncertainty by comparing our predictions to biomonitoring data. And when we quantify that, we can quantify the uncertainty on exposure and on toxicity and look at the bounds on those two items when we're comparing toxicity and exposure for risk. And that allows us to understand under what conditions we can still make decisions based on those values.
All right, thank you very much, and thanks again for your talk.
You're welcome.